Okay, cool. I don't.
There we go. Okay. So I'm here today with Michael Edward Johnson, who I've kind of recently become aware of his work, but he is a very interesting thinker. He previously did some writing on Qualia and Valence related to cognition and how those processes occur. And the idea that I'm most interested in today is he's written an excellent document called Principles of Vasocomputation, which we can get into a little bit more, but it sort of posits the idea that basically the neurovascular coupling, where smooth muscle that controls blood flow to neurons itself plays as much of a role in memory as connections between neurons, and that this is a sort of secondary layer of consciousness and intelligence and memory that isn't really isn't really addressed or factored in with some of our current models. So I guess maybe to start it out, my first question would just be, what kind of led you to that, or what was like what was the thing that sort of brought about that idea for you?
Yeah, that's a good question. So I would say maybe three threads. And the first was I was really I mean I'm just always very curious about how we work, what kind of thing we are. And it felt like I was sort of poking at this question of what is tension trying to do for us? And
Mm,
the sort of thought process there, like the train of thought was that um, we have an immune system, and if some bacteria or virus comes in, our immune system can fight it off. But computers also get software viruses, and it's like, what's the antivirus system for our software, for our patterns, for our neurons, we could say. And so I was thinking about sort of tension as a protective thing, not just physically, but also mentally, emotionally, cognitively, predictably. That sort of, if you had a bad pattern resonating in your nervous system, you could sort of tense to change the resonant profile and to sort of deaden the pattern. Um, so that was one of the three threads. And then the next thread was um, excuse me. I had a couple good friends tweeting a lot about Buddhist Tanha.
yeah,
Yeah,
this is really
yeah,
interesting.
yeah, super interesting. So Tanha is this concept that we try to control our sensations in ways that don't really make sense. So if a good sensation comes into the body, we just kind of reflexively try to grab it and hold it and keep it forever. And of course, this doesn't work. And likewise, if a bad sensation comes into the body, or like if we uh, dropped a glass and it shattered or whatever, we just very reflexively try to push it away and not feel it, to sort of deny the event. And likewise, this also doesn't work. And sort of the Buddhist frame is that um, the vast majority of our suffering is us trying to control our sensations in this way, not the actual sensations themselves. So, cool Buddhist uh, observation there. And then my question from a neuroscience perspective was how? Like how are we reflexively trying to do this? What
Yeah.
what system is kind of being leveraged here, and where is it? Um, and then the third thread was I was dating a girl who was very into sort of somatics and yoga and all this stuff, and I was just really curious, like, okay, what does it mean to have an open heart or a closed heart? Or, is this a real thing? If it's real, then it has some corresponding sort of hard physiology going on. And it'd be cool to dig into how this could be real or not. So those are the three threads that combined.
So maybe some questions that come up for me thinking about this. So another kind of the I guess the central topic it seems is the idea of like smooth muscle sort of having these three states that it can exist in of sort of like short-term contraction, um, relaxation, and then this long-term sort of fixed contraction or latching. And you sort of use this to illustrate like how blood flow might be sort of partitioned out of certain regions and into others. And so I guess I'm curious with this. Do you see this as because you've kind of said some things to the effect of like, well, maybe we could sort of unlearn certain patterns of like reinforced behavior, and we could open like a significant amount of latches, and what percentage of those could we open without sort of having negative effects? I'm curious if you see maybe the existence of something
Hmm.
like anti-latching regions. Like, do you see like lack of latching in a certain region being an enforced thing that occurs just as strongly as latching? Or do you see like latching in some cases as being a positive thing? Or are you thinking about it more in terms of like, well, we want the plasticity, the fluidity, so like long-term things are just restricting that. Um, so how do you think about it from that perspective?
Yeah, yeah, that's a good question. And maybe for the if we have listeners, I can also just kind of walk through Vasocom basics. That
Yeah, sure, definitely.
cool. Yeah, the basic grammar that Vasocomputation suggests is clench equals stabilize. So we have this muscle that's sort of distributed throughout our body that we don't even think about. That it's this layer of muscle, smooth muscle, wrapped around all our blood vessels. And from the big arteries to the mid-sized arterioles to the small capillaries. Um, and so vasocomputation says that when this muscle clenches, nearby neurons and sort of tissue modes, we can say, they stabilize. They don't change. Um, and I would, I guess, call out a couple sort of very important causal factors here.
Mm,
Um, one being, in a very simple sense, if we cut the blood flow to a region, it doesn't have the fuel to update. So it's very simple. You you got to have the metabolism going to in order to rewire synapses and
yeah,
whatnot.
definitely.
Um, and then the other is sort of the bioelectric uh, state of the latch. As these cells start to clench, gap junctions open up between these vascular muscle cells and sort of entrain a little neighborhood of cells with sort of the same, like they get electrically connected, I guess I would say, which kind of, I suspect, can sort of finely tune local modes in the tissue. So it's sort of this, like that's kind of the technical story. The more poetic story would be something like the hand of the mind is your blood vessels. Like that's how we can sort of stabilize patterns and grab ideas or release ideas. That's kind of the backstory. Thinking about latches specifically, this system can, because it's smooth muscle, it has this special setting called the latch bridge mechanism,
Mhm.
where normally you have the fibers, the actin and myosin that can sort of slide, and then a latch is when the muscle gets tight and then it basically glues itself together into a tight configuration. And you know, in the literature, this can last for a decent length of time, hours. I suspect that it can, in the body, uh, it can last much longer. So that's a kind of a note. And yeah, I think like to your question about like how does the body manage these latches? I see them as uh, predictions or Bayesian priors. Like every latch is sort of stabilizing a pattern or reducing the dynamism of local tissue. It's like putting the tissue in some sort of stasis. Um, and this can be used by the body for a lot of things. You know, probably on a daily basis, um, if we just like, oh man, I really have to remember to pick my daughter up from school at 3 or whatever, or like I'm gonna turn the stove on and I'm gonna walk away, but I'm totally not gonna forget it.
Hmm.
Probably we may implement these sort of medium-term memories, I would say, via vascular tension. Um, and then, once something comes true, like then you can just release the tension. But at the same time, I expect that we manage so much with the system, um, and that sometimes the system doesn't sort of do proper garbage collection, and
Mm.
we just kind of get stuck with old memory that maybe the body has sort of lost track of what it represents. Um, and likewise, if something is like very traumatic, if we're walking and get bitten by a big dog or something, then sort of patterns that would have kept us safe may get latched into our nervous system from that event.
So something that comes to mind for me here is I almost wonder if you've seen any connection between like let's say latches in a region as something that stimulates angiogenesis, because I almost wonder whether like during growth, for example, you could see like a more
Mm.
permanent mode of like latches as an intermediate to
Yep.
a state where you're going to have like vascular endothelial growth factor or something that's released, and then the the amount of blood flow to that region is sort of like permanently enhanced through that. Have you seen
Yeah.
anything with that?
So I would expect to see something like that. I think that it's a question of, I mean, first of all, like insofar as a latch is basically seen as damage to a region, then the body will sort of mobilize to route around the damage and
Okay,
grow more blood
interesting.
vessels. Or yeah, yeah, yeah. But
So
yeah, go ahead.
yeah, um, maybe my other question would be, do you see latches as increasing or decreasing blood flow, or can it
Right.
be either?
Yeah. Decreasing.
Hmm.
That and this gets into the muscle physiology stuff, but essentially, it's just like you can have blood vessels that are arterioles. Latches specifically um, are found at the scale of arterioles. So the not the smallest and not the biggest, but like the medium-sized highways of blood.
Oh, okay.
Um, the this like really tiny blood vessels have it's called pericytes. It's not exactly spelled the way you might think, but um, they're weaker versions of vascular smooth muscle cells, basically. Um, but yeah, I think that yeah, there's probably a lot there. Can you repeat your question, actually?
Um, yeah, I guess my thought process was along like whether we should think of latches as something that increases or decreases blood flow, because you used the word stabilizing earlier. So my maybe the question that I'm sort of getting at is, if we're stabilizing, how do you see that as occurring? Is it more so that the latches push blood flow out of one region and into another region that you're trying to stabilize, or is it that you're stabilizing sort of by subtraction, where you're like removing sort of I don't know, just to give a simple metaphor, let's say like region X represents like going up and petting a dog. Region Y represents like the other response. Maybe do you see it as like you're stabilizing one region, or are you taking out the other responses that you're trying to protect yourself from sort of going down those routes?
Yeah, yeah. So, I mean, I guess to restate your point, we have a finite amount of blood, and uh, tension in one area will sort of send blood elsewhere in the body. So it's like a balance thing. Um, I suspect that like latches in general are fairly they're blunt tools.
Hmm.
Um, and that I suspect the body might prefer to use unlatched tension in order to sort of manage like big flows of blood through the body, but not always. I think that like, but like the local effect of a latch will always be to cut circulation. Um, and like one interesting angle here is if we look at things like Raynaud's syndrome, that
Mm,
you know you see these crazy videos online where it's like the hand looks totally normal except like one finger or like half of the hand is just completely bone white. And I would call that probably more of a vasospasm than a latch, but vasospasms and latches are probably coupled, like causally, if there's one, there's more likely to be the other. So that would be like a an interesting example of like a latch we can see.
yeah. Okay. Um, so maybe another aspect to sort of look at here. I'm curious how much of a role you see this playing in like conscious memory. So there's sort of two things that inspired this. One was I was thinking about like reading about this. I was thinking about the case reports that come out of people that receive heart transplants, and they have
Right.
these like transfers of it doesn't seem to be like conscious memory, but it seems to be like personality characteristics or tendencies or like weird things like food cravings will be like implanted. And I think it's sort of inseparable because the heart, I think, is the second most neurally dense tissue in the body. So it's sort of like, it's hard to say, that this is just the vascular, this is just the neurons. Like it's obviously very interwoven. Um, but then the other thing, I was speaking to my parents about this. I was visiting them this week, and I was kind of talking about this conversation that we were going to have and some of my thought processes. And my dad had the question basically. So if you were to have like an amputee, um, like do they lose memory? And I was
Right.
like, maybe that's not the right way to think about it, but maybe it is. What if there is something to that? And I'm curious, kind of what your response to that would be.
Yeah, yeah. I mean, super interesting questions and super important, I think, too, in sort of strange ways. I do think that I mean, one thing that I'd point at with the heart is that it's like people think of the heart as a pump, and it kind of, that's one of the things that it is. But I think it's sort of um, a global, like centralized coordinator for a lot of
Mm,
things in the body. And like the sort of as like an electromagnetic metronome and whatnot. I think there's really something there. So if you replace a centrally coordinating node in your body, you should expect some patterns to transfer over as well. And
yeah, definitely.
I've heard, read these stories about memories and stuff, and it's really wild. Um, I I expect that some of that could be, not necessarily neural memories that I mean the heart has something like 40,000 neurons or so, but is like very, very dense with neurons. Um, but in terms of uh, what priors are really encoded in the heartbeat, once you think of it as, not only a mechanical thing, but an electromagnetic thing and so on, like there's probably a lot of information that we just don't know yet about that. Um, so I guess short answer, I think it's plausible that like the memory stuff.
Yeah, definitely. Um, so this kind of one of my big things over the years, like I've just moved more and more in this direction. Is like there's no hard division between like neurons and the brain. Like I think that the way we use language a lot of the time is limiting to our ability to see things as they actually are. And
Yeah.
a lot of this comes in with like things like neurotransmitters with hormones, with neuroscience. Like um, sort of my mantra at this point is like everything is a drug, everything is a receptor. It's like there's no hard cutoff where like something doesn't affect everything in the body. And so
Yeah.
like from that standpoint, um, I really start to think about it as like, well, every cell behaves like a neuron to some degree, and we see this like really clearly. Like most cells produce neurotransmitters like glutamate and GABA, and they respond to them in the same way that a neuron would. We even see like the neural growth factors like BDNF is produced in other parts of the body. And something else I kind of wanted to see if you had any thoughts on is like platelets. There's a really interesting paper that basically suggests that like platelets are a proxy for neural activity because they can carry like an imprint of neurotransmitter levels in the brain in circulation.
Mm.
And so, like, for example, there's some research suggesting that autism has higher levels of circulating serotonin and platelets. And that's both an imprint of like gut serotonin synthesis because the serotonin transporters from the gut endothelial tissue basically offloads excess serotonin into platelets. But then it can also be an imprint of neurons, basically in a similar way. And so I'm curious if you see any like circulating mediators having an influence on latches, or I guess where you see that coming in from. Is it from like the neural side? Is it from the blood flow side, or both maybe?
Yeah. Yeah, that's a good question. And I would say that one of the most interesting answers that I've found to this is actually carbon dioxide. That
Yes.
very, very peaty, of course.
Yeah.
Um, and so I mean, part of the hypothesis about like what I think breath work is doing and how it does what it does is if you're like, and there are various forms of breath work and so on. If you are hyperventilating and you're sort of dumping a lot of CO2 through your breath, then the body has to mobilize its CO2 stores, from bones and whatnot, and then that gets circulating in your blood. And CO2 is one of, if not the core, vasodilator.
Mhm.
Um, and I suspect that the sort of extreme opening events that people get with breath work may be in fact due to jacking up the CO2 in their blood that way.
Mm. So this gets into a lot of interesting stuff. So um, one thing that I really like with um, with with CO2 is that it sort of has this role as like almost intelligence when it comes to being a signal that different proteins respond to. Like it's something that you see a lot of the time, and we can get into this a little bit more later. This is something else I wanted to talk about, but proteins have intelligence and they respond to different things in ways that are um, very fine tuned to their environment. And so we have this thing of like the Bohr effect, where hemoglobin sort of has a certain amount of CO2 present, and that it senses pH and has structural interactions with CO2 that cause it to release oxygen more. And you could see that
Mm.
as if hemoglobin's sort of goal is to get oxygen into tissue that needs it, then CO2 might be a marker of that, of higher metabolism. Um, the other thing that this really intersects with for me is post-translational modification, which is the idea that proteins sort of once they're initially synthesized, they have a life that they go through, and they sort of pick up these different structural changes over time. Because if we think about the way that proteins are structured, sort of each amino acid in a protein has bare minimum carboxyl CO2 group and a amino NH2 group, and those can sort of combine to form peptide bonds. But between each of those groups, there's going to be like a sort of middle structure that's these different side chains, and the side chains just mediate all of these really diverse interactions. And so I know we're both fans of Gilbert Ling. He writes a lot about these sidechain interactions with the charged amino and carboxyl side change, where they're either negatively or positively charged. And these can form like sort of opposite charge linkages that mediate things like the coiling and folding of proteins. But there's also a lot of cases where you see basically the side chains can become like modified, like they can sort of have attachments to them.
Mm.
And one of these is CO2. Another one that's really interesting to me is nitric oxide. Um, I like to use a term, nitric oxide pooling, basically, which
Mm.
is my
Yep.
it's sort of me sort of playing with language because you like to sort of think of these things as like being very short acting mediators, at least from like biochemist perspective. It's like nitric oxide is almost like a free radical where it behaves very quickly. And
Yep.
because of that, sort of the idea that it could stick around and like float around in tissue is like very counterintuitive. But I think of the way that it interacts with proteins being almost like a sponge, where like a sponge or a piece of cloth has these fibers, which ironically, if it's organic, are also
Mm.
proteins, and they interact with water in such a way where they form basically bonds with their surface area that cause the water to be retained, and different forces that you can exert on that can cause the water to be expressed like pressure. And
Mm.
so I sort of think of it in the same way of like you can have like an imprint that's left on different proteins based on the
Yep.
presence of things like CO2 and nitric oxide that sort of is like an echo or a lasting imprint over time. And whereas nitric oxide is like milliseconds or these very short time scales that it acts on, the imprint can last potentially hours or longer. Very similar kind of to like a lower level of the idea of like short-term contraction versus latching.
Yep.
And so one thing that's really unusual, and I kind of disagree with Ray Peat on this, is that he sort of looked at it as like there was CO2, which is like the good vasodilator, and there's nitric oxide, which is like a bad vasodilator. And these are like he liked to view things in this polarity of like these are the mediators of disease, these are the mediators of
Mm.
metabolism and health.
Yep.
But it's not so simple because when you look at how CO2 dilates blood vessels, it actually does that via locally increasing nitric oxide. And part of the way it does that, in my interpretation, is by increasing oxygen availability for nitric
Mm.
oxide to be synthesized from oxygen. And so,
Got it.
um, one of my thoughts with reading through your work and kind of thinking about like nitric oxide pooling and my own ideas is that maybe there's like a sort of form of anti-latching that could occur with nitric oxide pooling in vascular smooth muscle. Because I was kind of looking at this like, well, if you have nitrosylation occurring in excess in a smooth muscle cell, does that predispose it to latching or to predispose it to not being able to latch? And it's basically the latter of the two, that it doesn't allow latching to occur, and it would sort of perpetuate like a long-term relaxation. And
Yeah.
so I almost wonder, in cases of like inflammation, for example, where nitric oxide is also mediating this like immune response to infection and things like that, whether we could have anti-latching that sort of destabilizes the system in a way that could be like an opposite polarity almost to like latching.
Mm. Interesting. Yeah. It makes sense. I think that I'm reminded of I would credit anabology for this. I haven't actually made the beam, but I'm wanting to. It's like the his uh explanation of the peat approach to different like substances is you have the mid-wit curve, and
Mm,
at the bottom it's like something's good, something's bad. And then the
yeah.
midwit it's like, well, it's all very contextual and just a wall of text. And then the top wit is, yeah, something's good, other things bad.
Yeah.
But maybe there's more to it. I'm very sympathetic to this idea that like the body has reasons for what it does,
Hmm.
and so I'm hesitant to say NO bad, CO2 good.
Mm,
Uh, I I I'm familiar with a little bit about your NO pooling stuff. I definitely want to know more. And I think that I mean it all also reminds me of this is also this dance around this central property of energy. And like a latch is, in some sense, a latch is a clench the body doesn't have to pay for. It's sort
Mm.
of you can set it and you can forget it, and then you basically have to feed that tissue less. You don't have to send it as much nutrients because you can't. It's sort of a way to arbitrage energy demands in the body. Um, and then, as to your point about inflammation, it is possible that, okay, the body can become miscalibrated in terms of like what to keep latched and what to not keep latched. And like we could think about the manifestations of this. But I do think of this as kind of like energy arbitrage is, I think, a very important part of this. And like I'm um I think that people probably underestimate the role of metabolism in
Mm.
releasing tension. So
Yeah.
I'm reminded of like Gerald Pollack has sort of taken a lot of Albert St. Georgi and Ling's work and sort of had the constructed this model of neurons and muscle cells where it's like the relaxed state is the high energy state. It's like the nicely ordered low entropy state, kind of pushing the ball up the hill or the stone up the hill. And then once you have an ordered cell, then you can perturb it. And then ball just kind of rolls down on its own and the muscle just kind of contracts on its own. Um, but like you actually have to pump a lot of energy into cells to relax them.
Mm.
And like I don't know if you've ever had the experience of, you're laying in bed after, a completely exhausting day and you're literally too exhausted to sleep. You're too
Yeah.
exhausted to relax. And the like the bioenergetic perspective is like, yeah, it makes sense. It takes energy to relax your muscles. And if you don't have the energy, then they're not going to relax. So I think that like delatching is going to be an energetic thing along with everything else. Um, and I mean, I had, it was like 70% joke, but 70%, maybe not 90% serious. A tweet that, maybe the extreme rarity of classical Buddhist enlightenment today may be due to basically everyone being mildly hypothyroid.
Mm. Interesting.
Um,
So
yeah. Okay, yeah, go ahead.
yeah, so my thought process in this, one of my favorite papers like this past year or two has been it's called ATP as a biological hydrotrope. And
Cool.
it is like it intersects so well with Ling. And because I think it explains in like a different sort of vocabulary than what Ling was getting at, of like
Right.
this idea of, because he sort of has this idea of like the resting state versus the active state of cells. And
Yep.
I think when you really start to look at this, it's a little bit more granular than that, where probably there's like regions of active state versus resting state. And he sort of has this idea for this of the protoplasm, sort of reviving this term of protoplasm and thinking of individual proteins as units of nanoprotoplasm, sort of like the
Yep.
building blocks or the pixels or the atoms of this.
NPUs and yep.
Yeah, yep. And so um, sort of thinking about it from that perspective, you can sort of get some of these together. And when they are in unfolded states or folded states, then they can accomplish different things. And somebody that Ling was sort of inspired by or worked around the same time period as a little bit um, was Dmitry Nasanov, who was the Russian colloid. He was like a biophysiologist from the Russian Nasanov school, and he basically thought about things in the sense of proteins are denatured by excitation, and for an individual protein that might mean something different for another. So if the protein is natively folded, then denaturation would mean it's unfolded to be sort of turned on or excited. And then other proteins that are natively unfolded, they might fold when they're when they're turned on. So it's sort of like this shift from one state to another that's not necessarily always going to be folded or unfolded, but he he made sort of early observations that cells display more turbidity. They're almost more like cloudy or have like regions of opacity that form when they are excited. And this is like you can measure
Hmm.
this with different optical tools. And so um, he found that high degrees of excitation basically led to viscosity and like very rigid structure within the cell. And that there was a point of irreversible excitation where that simulation would become like overly viscous and rigid.
Hmm.
Um, and the interesting thing is this relates a lot to ATP because ATP, I referenced that paper, ATP as a hydrotrope. A hydrotrope is almost like a detergent. So, like the way that soap works is that it has this sort of charged hydrophilic head group, and then it's got a tail. And so, like when we use sodium hydroxide to make it, it's the sodium that basically becomes the charged head, and then you've got this lipid tail. And so, sort of the if you have like a fat soluble like oil or something that's gotten on your skin and you try to wash it off with water, it won't come off. But if you wash it with soap, then it sort of orients in a way where the lipid soluble part of the soap will orient toward it, and then it leaves like a hydrophilic exterior that allows it to form these like particles that can clear it. And so ATP has a similar structure where the it has this sort of like combination of regions of like hydrophilic and hydrophobic throughout its structure, and that allows it to work similarly because um, the adenosine kind of end, the base, and then you have the phosphate tail attached to that. The adenosine base is able to basically have these interactions with hydrophobic regions. And then the phosphates sort of like shield those regions from each other. And that
Hmm.
prevents them from sort of clumping together and folding the protein into itself. Um, and then when you have ATP that gets broken down or mobilized, those regions get unshielded. And this is there's a term for this. I don't remember it off the top of my head, but it's basically entropic protein folding or um, something along those lines. It's basically that the hydrophobic regions seek like a lower energy state, and so they'll tend to like fold in on themselves, leaving the hydrophilic regions exposed to the outside. And ATP sort of like unfolds that geometry by shielding a lot of that. And Ling sort of writes about potassium being similar, and so kind of makes me think about from a standpoint of like latching or muscular contraction, whether enough ATP sort of predisposes to less latching because of that sort
Hmm.
of like you have the energy to shift between those two states. And like I really like the way you were talking about it from the standpoint of like budgeting energy. Like if we're trying to say let's avoid ATP expenditure, well, if we're keep contracting and uncontracting and sort of ATP is running low, we either end up in a state of like excitation to the point of permanent denaturation and sort of Nasanov's terminology, or you end up with a sort of permanent fix that can just say, let's leave this alone. We're not going to worry about it for a while and we'll come back to it when we have more energy.
Yep. Nice. Yeah, I think there's probably a lot of that. And it's like I would also say that just sort of anecdotally or in terms of there's a strong trope that I think is true. Um, Fasting is a very interesting scenario to think about here also.
Mm,
Um, like obviously the body sort of shuts down progressive systems during a fast, and um, I think that I'm I'm honestly really deeply curious about if you could make a map of latches through the body, and like version 2 or version 3 of the Midjourney ultrasound scanner, something like
mm,
that.
yeah.
Um, yeah. Then, if you did it, every day of a fast, what would happen? Would you like, I feel like you might get um, more latches in some regions, but maybe less in others. And then I think basically the refeeding window, I think you'd get a lot of latches releasing.
I
You
can
just get
definitely
this,
see that. Yeah.
yeah, this flood of energy, and the tissue says, okay, well, kind of forgot what we're doing, but anyway, we have energy now, so let's do it.
Yeah, I experience that a lot personally with like calories have like a profound relaxing effect for me. Like I
Yeah.
really find that like when I have like a conversation like this or when I'm doing a lot of like deep reading or something where like my mind is really active and then afterward it's like you said almost like you're too tired to relax. Like you just want to lay on the floor and just kind of take a moment to collect yourself. And like food fixes it very well. Like just having some like X amount of calories is almost enough to like reset the system.
Yeah.
And it's like um it's something that I think Pete writes about that really I resonate with. It's just this aspect of sugar and carbohydrates and things like that having this relaxing effect. And I think it's unfortunate because we're in a period of sort of like the way our food and food availability is structured that sort of skews toward those things having their own pitfalls now because
Yep.
they're often integrated into like ingredients that are toxic and things like that. But um, in the purest form, it's like you really would want those things to be like the quickest way to replenish your energy and regain that stasis. So that's really interesting to me. Something else that I've been thinking about with this is serotonin,
Mm.
because I don't know how familiar you are with Chris Masterjohn, but he has some really excellent
Yeah.
stuff on serotonin. And something that I've picked up from him that I really like is he reframes it from the perspective of hypoxia. And basically, sort of, we can think of serotonin as something that's meant to respond to hypoxia in a contextual way in each tissue. So, in the digestive system, serotonin stimulates motility and contraction. It sort of, or in the upper intestine, it stimulates nausea and vomiting. And
Yep.
so, the reason for that might be that we have this sort of limited blood flow to the digestive system. And if you were to have like a buildup of material in the gut that's not moving, then it's exerting pressure and further restricting that blood flow. And so you want to reduce the possibility of necrosis or damage to that tissue through hypoxia. And so serotonin gets released under those conditions of like stress and pressure and injury, where it's sort of saying, like, get whatever is exerting this pressure out of here, whether it's from the
Yep.
top or from the bottom. It's just like move it away from this space. And
Yep.
in the lungs, you see a sort of different role where um, serotonin is sort of doing something similar where regions that have low oxygen saturation will have serotonin be released and it constricts the blood vessels in that region to sort of maintain more blood flow in the regions that are getting oxygen. So it's like push the blood flow out of here. We can live without it for now. It's better that
Yep.
we get the most oxygen possible. And so it's sort of like exerting pressure away from it. Um, and so kind of intersecting back with a couple other points here. Um, Serotonin can undergo post translate or can be a substrate for post translational modification, similar to CO2 and nitric oxide and some of these other things. So you can have serotonylation of proteins, where you can have like a lasting imprint of serotonin that is exerted on different tissues. So that's interesting. Um, and then beyond that, I think something that I've been thinking about that I was sort of happy to find that there's some other people thinking along the same lines is I had this idea of, well, what if psychedelics? Because things like psilocin are very, very structurally close to serotonin. Like it's
Yep.
basically serotonin if you used the dimethyltryptamine as a base instead. So it's got the methyl groups on the nitrogen atom, and then the OH group is like shifted one position over. But otherwise, it's like very similar. And so I'm thinking like, well, if you take off one of those methyl groups or two of those methyl groups, now it can be post translationally modified onto proteins in the same way that serotonin could. And so
Fascinating.
yes, this is like
Uh,
a very exciting thought for me. And the cool thing is, I think that maybe the hydroxyl group being shifted over closer to the nitrogen atom is important as well, because that could lead to some hydrogen bonding interactions with the protein that it's interfacing with that could make it more stable long term.
mhm.
Um,
Fascinating.
And so I couldn't find any experiments with psilocin looking at this, but I did find there's a group. This is like a preprint. It's not published yet, but they made a mescaline analog that would respond to this particular assay. And they did like a high dose, and they added some extra calcium to kind of facilitate like post-translational modification with this mescaline analog. And they found that like dozens and dozens of proteins within the cell responded to post-translational modification by mescaline. And
Interesting.
a bunch of them are related to energy metabolism, and a bunch of them are cytoskeletal proteins. So there's some very cool
Oh,
stuff there that I don't know the full implications of. And they sort of give the caveat that like we really pushed it in this direction, and maybe only a fraction of these are actually going to respond this way under normal conditions. But just the idea that the afterglow and things like that of psychedelics could be exerted by post-translational modification. And then the other way that I've been thinking about this is if we look at the fact that these act on serotonin receptors, maybe we can think of psychedelics as sort of flexing the hypoxia system, like they exert this temporary hypoxia response broadly across the whole system. And then there's more resilience to these hypoxia responses afterward as a result
hmm.
of this sort of like almost flexing all the muscles to train them in a short period of time. And then once they're relaxed, they're sort of more fit to
Yep.
carry out hypoxia responses afterward.
Interesting. So, a few threads here.
Yeah,
I mean, that's fascinating.
yeah,
Yeah. I think that, I've been thinking about something like psychedelics as very effective at sort of stochastically opening latches. And, there are various sort of views on how could they be sort of driving wave-like behavior in the vasculature and so on. But I think like the hypoxia angle really has a lot going for it, and I do think that you know what 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 is the local logic of a latch when there's a global hypoxia event?
mm-hmm.
Maybe it turns out to be an open signal, and it would be interesting to sort of trace that through. Um, I'm also and just kind of a slight detour back to like an interesting sort of expression of this. I think that there's as a writer, as a theorist, I'm always doing a lot of deep work, and it takes me a long time to sort of release an idea and like release the context and sort of empty my mind for the next idea. And
Mm,
I was thinking a lot about this, and in terms of if if sort of the micro pattern of tension across your body sort of sets the context for the your problem domain. If you're doing math, it's like you kind of program your nervous system such that like your sort of the resonant profile of your patterns fit the domain, we can say. And then, if you move on to a new program, well, you have to reprogram all this micro tension and sort of learn how to do that. And I've been thinking a lot about how like the extreme downtime needs of creative people might be sort of more muscular and metabolic than we realize.
mm,
And I wonder if a creative wants to sort of, you know, move more quickly, then like maybe like do more peating and do more sauna and it's like
yeah.
approaching it as an energetics problem might be very interesting. Um, so, but any like back to this like imprinting thing, I think that I mean I don't know much about the system. A lot of the stuff I'm hearing for the first time, so
Yeah.
I I love it. And yeah, I guess I'm really wondering what what does it mean? Like what kinds of molecules can imprint? If mescaline and serotonin and NO, CO2,
Hmm.
psychedelics, if these can imprint, then like can we generalize these molecules of or these classes can imprint, but these classes can't?
Yeah, it's like a really broad actually
Hmm.
category.
Yeah.
Some of like there's some excellent papers on this. I'll see if I can find one and send it to you. That's just like a good overview because it's like um, most neurotransmitters, dopamine, serotonin. There's a group that proposed the term amphetamine for amphetamines being like similar enough in structure to dopamine. Um, there's some interesting stuff with that too in addiction. You see basically an increase in dopamine that's measurable in addiction. So you see like the imprint of dopamine on proteins in the brains of addicted mice or humans or things like that. Um, Histamine can imprint. Lactate can imprint. So under conditions where like maybe hypoxia or like um, I think of it more in the terms of like shifting from pyruvate to lactate as like it's like a response to the redox state, like the electron balance being overly saturated because there's not enough oxygen to like
Mm.
pick it up. Um, and so under that condition, sort of like the electrons in the electron transport chain get backed up and you get the shift in things like NADH to NAD plus ratio that's often like talked about in aging. Um, and then basically producing lactate from glucose creates NAD plus. And so there's all these processes that are sort of like release valves for electrons
Right.
that free up the cofactors. And one of my big things that I'm a fan of is the use of quinones. Um, Pete writes about this a little bit as well with like vitamin K, CoQ10, but there's also things like black seed oil, black cumin oil, which contains compounds like thymoquinone. And there's some other kind of like PD things like Cascara sagrada and things like that, which contain quinones. And these also, in the process of being like detoxed within cells, they also produce NAD plus. And so there's
Mm.
this sort of further ability to like um, free up electrons. And then beyond that, you have like glutathione, post-translational modifications. You have there's things like ubiquitin and things that are used in sort of
Mm.
like tagging and regulation. Um, and then there's even cases where like larger proteins can be imprinted, or lipids is another one that's really interesting. So, for example, with like fatty liver, things like that, you might have like an imprint of palmitate, like one of the long chain saturated fats could be
Mm.
complex proteins. And all of these are going to have their own effects. Like I'd imagine, or I've seen a little bit to suggest that lactylation can induce different hypoxia response things because it's like the increase in lactate has these different impacts that you'd associate with hypoxia or excess electron saturation. And so then you're going to want to respond to that. And so sort of proteins have adapted over time to like read these as environmental signals that create like memory and how they respond to their environment. Um,
Fascinating.
yeah, there's some really interesting stuff with this. And um, maybe this is a good time to bring in another paper that I just found this week that is absolutely excellent. Um, it's called Intelligent Proteins, and um, it's by Tripathi et al. And this is another sort of thought process I've been thinking along with the lines of both Gilbert Ling and Michael Levin's work has been really inspiring to me in this realm as well. Um, Sort of Gilbert Ling talks about nanoprotoplasm, like these protein units having their own sort of like responses, active states and resting states and things like that. And then this being like the basic unit of life. Like he titles one of his books like Life at the Cell and Below Cell Level. And he
Yep.
literally means life below the cell level. You know, it's like very interesting. And then Michael Levin sort of talks about these structures or patterns having like agency and carrying out sort of different like goal directed behaviors. And so I was looking at this and I was thinking about it from Gilbert Link's perspective. And it's like, if you look at the average number of amino acids in a protein, it has 300 to 400 different amino acids in a protein. Some of them have 1,000. And if you look at the number of chemical bonds in the average protein, it's like 4,000, 5,000, 6,000 more. And each of those bonds can undergo rotation, it can go flexion, it can go stretching symmetrically and asymmetrically, and it can translate through space and move in different ways. And when you sort of stack all that together, you have this enormous range of like possible shapes that a protein can take. And obviously, some of those are going to be restricted by like interactions with itself. But it's almost to me like a computing system where you can sort of think of it as like different conformations are creating different outputs and things like that. And so my initial thought was, well, maybe we can consider each protein as having goals that have sort of evolved over time, and it's reading inputs from its environment, um, and it's putting out different outputs based on that. And there's sort of these complex things. And with something like a receptor, for example, you can have it read something completely novel, like a drug or a pharmaceutical or something that humans design that it's never seen before. And it's going to read that and try to approximate it to something that it knows. Like
Yep.
if it reads a psychedelic, it's going to say, well, this is kind of like serotonin. So I'm going to respond kind of like the way that I would respond to serotonin, but it's got elements that make it sort of like almost the opposite of serotonin. So I'm going to do certain things that are different because some of these things like psychedelics are partial agonists on some of these sites. So they have like almost a mix of activity, and it's like it's reading both properties and responding to them under different conditions. And that's incredibly
Nice.
interesting just on its own. But then when you bring in this idea of post-translational modification and like imprinting, um, it's as if it stores memory. And we see this as well. And so in this paper, they outline like some of the different ways that proteins can form memories and learn under different conditions. And for example, they use the example of kinesin, which is like a motor transport protein that walks different cargo along microtubules and these cytoskeletal proteins. Everybody's seen like the animated video of it like carrying the cargo. Um, and so this is actually like a very dynamic process. It'll vary like the length of its steps and the rate of its steps depending on the cargo and like the size of what it's carrying. It can tag microtubules and put down
Mm,
markers so that other kinesins will follow the same pathways, or it can tag microtubules so that they get broken down after it's gone through them. It
mm,
can basically modify its environment in a complex way. And if you were to describe this to a child and say like, well, there's this little thing inside you, and it's making decisions based on what it feels, and it's putting down signs in its environment and things like that, they would think of it like, oh, there's a little creature that lives inside me that has its own sort of life going on. But we are totally reducing this down to some like mechanical process. We're saying, oh, this is just like a machine and it doesn't have any intelligence or thoughts of its own. So I don't know if you have
right.
any thoughts based on that, but that's just something that's been fascinating to me recently.
Nice. Yeah, 100%. I mean, there's so much intelligence in these systems, and I'm reminded of Gilbert Ling's sort of frame of the cell as an electric computer,
Mm-hmm.
and I've been thinking a lot about like, well, how do you program this electric computer, and kind of the like exactly what happens when a receptor gets activated. And I believe Ling's view was that yeah, you can kind of adjust the sort of the local tightness of the field in certain pockets by sort of
Yeah.
depositing electrons or withdrawing electrons. And but yeah, I mean, biology is going to use all the tricks, and I like the sort of protein as a computer frame as well.
Yeah.
Um, I mean, I'm also reminded of just how like there'll be like one motif and it'll be copied all over the body. And
Mm-hmm.
thinking a lot about serotonin. Um, One of the most interesting uh, theories I've seen about the grammar of neurotransmitters, we can say, is that um, you know basically the body took the basic situation with the gut, and you have kind of this metabolic process which sort of extracts all the good stuff and then excretes the waste. And depending on whether it's like a good tasty thing you want to like spend time extracting from, or whether it's kind of noxious and you just want it out,
Mm-hmm.
serotonin may sort of increase the frothiness of the um, peristalsis patterns. Um, and likewise, like the theory goes, maybe we sort of took the same template and use it in the brain. That instead of food, we're digesting sensations. Uh, and we sort of vascular muscle. It doesn't exactly have peristalsis, but it doesn't not have it either. It's like there are some interesting oscillatory dynamics there. And you know, if serotonin is like it's often framed as like the coping hormone, and there's active coping and there's passive coping and so on. But basically, like if you just want to sort of, it's also sort of uh, can be a very like coupled with dissociation.
Mhm.
Um,
Yeah.
a- and so if you have something noxious and your body is just like, we don't want to metabolize it, we just metabolize it, we just want to get rid of it. I could see a serotonin dump as sort of filling that same functional role uh, as it does in the gut. So, I mean, super interesting. Okay, can we do this with all hormones and like in what
Yeah.
way do
Right.
we metabolize? Yeah, I mean, the list of ways we could go here is huge, but I always thought that was a fun model.
So something else maybe I wanted to touch on a little bit here. And this I'll say like I know the least about this probably out of your work. Like I just didn't have time to review this in depth, but you have the symmetry theory of valence. And um, so maybe some thought processes just hearing a little bit about you speak about this with other people.
Yep,
It's, I wonder how you see this like on what sort of frame you see the symmetry or on different scales, maybe how that symmetry plays out. Because something that I was in this intelligent proteins paper, something that came up with this is basically that proteins exhibit hysteresis, which is like they take different paths back to the resting state depending on
yep,
their experiences, and that can be sort of considered like a marker of memory. Is like the protein goes into the active state, it has some post-translational modification or some structural change that occurs to it, and it goes back to the resting state. But because it has this change, it can't take the same path it took just in reverse. It has to sort of slightly alter that path or adjust it to reach that. And from the standpoint of that idea of symmetry,
yep,
that to me sounds like a symmetry break, but maybe that's not the case because um, maybe you don't think about it in terms of like temporal interactions where like symmetry um, does or doesn't occur through time. So maybe it's more spatial. So I'm curious, like, is this something you
yeah,
see as the cellular level, or does it really show up more at the organism level? Like you use the
yeah,
analogy of a starfish having the symmetry of like the five arms, and if one of the arms is damaged, then that's loss of that symmetry. And so it's going to respond to that as a negative stimulus because of the symmetry being lost. But
yep.
do you see that as extending down like very small levels to proteins or cells, or I guess where does that kind of show up the most?
Yeah. Yeah, good question. I would say that when all is said and done, I think it'll show up even more strongly in cells than at the organism level. Um,
Interesting.
and so, like, I give the example of sort of symmetry as sort of constituting or like uh, uh, constituting a system's attractor, and almost like a disorganized system can sort of follow the symmetries back to a state of order, kind of like find the kaleidoscopic grooves and sort of fit in to be whole again. Um, and just like symmetry is just a very powerful principle of self-organization, and systems are just going to use it. Um, I do think that like I I've been thinking, and just to step back a little bit for possible audience,
Yeah.
the symmetry theory of valence is a theory about pain and pleasure. And it's this thesis that if we had a perfect mathematical representation of an experience, like what it feels like to be me right at this instant or you, then we could sort of take a look at this mathematical object and we can think of it like a very high dimensional shape. And the pattern or property that represents, and like everything about our experience is in here somewhere.
Mm,
And the pattern or property that represents how pleasant this experience is would be its symmetry, um, which is kind of a fancy formal way of saying harmony in the mind is what feels good. Um, but then there's this question of, okay, like, can we apply, some of these lenses to cells? And I mean, my answer is absolutely yes.
yeah,
Uh, and I expect that, I guess in sort of looking at what's likely to happen across the next few years, I do expect that, like, I mean, everyone's trying to build a cell foundation model of like how to like measure a bunch of stuff and see like do next token prediction on cell evolution, we can say. Um, and I think this is a super important question and super hard question. And it's like obviously, you need a lot of data, but you also need to be a little bit opinionated about what you measure in a cell. It's not just like gather raw data. There's just so many degrees of freedom about what to even pay attention to. Um,
yeah,
and so I think that like a symmetry foundation model for biophysics makes a lot of sense to me. Of like, okay, there are going to be implicit conservation laws in in a cell, in cellular evolution, in cellular configuration. And can we extract them with machine learning? Can we sort of, use these lenses of like symmetry and gauge theory and whatnot? I mean, in one sense, all of chemistry is just accounting for what's happening in the EM field. Um, and like it's a great system of accounting, but like we can really lean into that and sort of yeah, I have a lot of thoughts here.
yeah,
But yeah, it's I just want to point at it as this is a really cool problem.
yeah. So maybe so one of the ways that I've been thinking about this idea of like intelligence in proteins is that um, they sort of are exploring a space of like possible configurations and sort of like there's paths that they are more likely to follow than others. And so
Yep,
maybe the way that I would interpret this from what I'm hearing is like that maybe it's better in some sense to view it as like this space of possibilities displaying symmetries rather than
yep,
like physical structure necessarily having symmetry. Like sometimes they correspond, but you can have like an asymmetrical organism or something and still display this. Though obviously, even within organisms, like they do display a remarkable degree of symmetry. So I don't know. There's some interesting stuff with that.
yeah, yeah. I think many ways to go, and it's just like yeah, I guess like I just point back to this question of like what will the principles of the first sort of successful cell foundation model, like what will it use, and and like what could you do with it? And I think the sky's the limit there.
Yeah, yeah. Sort of on that note, the something that I've been also thinking about with regard to like the way that we model cells is I think that really it'll be interesting to see the movement in the direction of these like um, electric sort of field effects because
Yep,
I think there's a strong tendency to try to model cells from the standpoint of like, well, this enzyme has an affinity for this, and in the presence of this, it's going to have like this sort of chance of occurring with a reaction. And if we put them together, they're just kind of going to like bounce around through Brownian motion until they like just randomly interact.
right.
Um, but there's this sort of lineage of theories that sort of goes through this other side of the equation that's like, well, what if they exert like long range effects on each other?
Hmm.
So there's at least two that I really like here. Irina Kosick. I'm not sure if you've seen her work, but she has the something called the resonance recognition model, which is really interesting. So she suggests basically that I think she interprets this as like collective resonance across an entire molecule, but maybe I would, and if I could be wrong on that, but I think maybe I would think of it more as like local resonance of like a reaction site, sort of
Hmm.
having a certain frequency that corresponds um, to a substrate. And so the substrate's going to resonate at the same frequency while something that's not a substrate won't. And so they'll actually be drawn together through some sort of like long range interaction like that. Um,
Hmm.
Another one that's sort of more well established and accepted is the biochemist Area Warshel has a theory that he calls electrostatic preorganization, which is basically that um, there's an electric field, like even just a static field put out by a protein's sort of reaction site, and that can actually shift the state of a substrate into a state that's sort of more favorable. So it can sort of like push the molecule into like a transition state before it actually touches the enzyme. And so
Well,
it's sort of like we have these enzymes with incredible efficiency. Like they can even approach perfect efficiency in some biological enzymes. And it's like when we do synthetic enzyme design, we can't quite get that. Like, and it's sort of like a question of like, well, what are we doing wrong? And I think it's that we are not fully factoring in these like electrostatic sort of like attractions and pre-organizing influences where it's sort of like, well, let's move this electron over here because we have more of a positive charge in this region and more of a negative charge here, and that's going to sort of prime it for the reaction. So there's some really interesting stuff there as well.
yeah, fascinating. Yeah, I think that I mean, I'm really curious about looking at the big AI labs right now, Anthropic and OpenAI, and they're trying to go full speed into biology, and like they're gonna run into some of the stuff, and there might be some surprises.
Yeah, it's like sooner rather than later we're going to have to reckon with like how like where the gaps are basically. Like
Yeah,
we don't understand why this isn't working, we're like let's go hunt for some more theories, and maybe this is where like people like Gilbert Ling um sort of come back into the picture. Like it all comes full circle and we're like, well, I guess we have to address the elephant in the room, which is that this sort of colloid chemistry or adsorption of ions or electrostatic interactions or whatever, that these have been at play the whole time.
yeah, 100%. I mean, I've sort of come at this in general from the perspective of I want to understand consciousness. I want to understand the mind and so on. And this very naturally sort of leads one into, neuroscience and then biology and then cell bio and whatnot. And it all matters. And
Mm.
then it's like in order to compile it back into, kind of a clean theory of sensations or whatnot, qualia, then you really do have to get the details right. So yeah, I'm thinking a lot about cell bio these days, and specifically like what happens to local tissue when there is a latch. Kind of to bring back that topic that there's this it's it's like a fascinating study in, I'm going to say, bioelectricity,
Hmm.
where you have these and need to shout out to my friend Nick Ford here. He helped me think through the gap junction stuff. And so yeah, you have kind of almost a bioelectric pocket universe where you have normal tissue and then it clenches and latches, and then you have such strong boundary conditions that there's like a very clear inside versus outside. And there's not actually a lot of information coming in or nutrients coming in, or sometimes waste going out. And so you have this sort of like little pocket in the body or like little cyst. And I don't know, it almost feels like a seed of disease or like a seed of cancer, that you have basically all the ingredients there. And I think that understanding, okay, what's actually going on in the cells, like does it collapse the metabolic repertoire? Does it like how big is this pocket? uh, like a latch might be 100 micrometers to 200 micrometers, depending on tissue and context and whatnot. But yeah, I guess I'm somewhat bullish on latches as a really interesting intervention point for for studying cell bio. Um, you know, even regardless of like kind of the somatic, yeah, it feels good to open latches and stuff, but like it feels like it's almost like this is a way, one of the crucial ways the body sort of changes the fundamental equations of like what the tissue is in a reversible way. So yeah, I don't know if that connects exactly to what we were talking about, but it
Yeah, no, I
feels cool.
maybe comes full circle to something else I wanted to pick your brain on is you
Cool.
mentioned that um, excuse me, you mentioned that basically in kind of our messages before this um, that there's some general trends like that you said something about basically that most latches open under warm conditions and something that I was thinking about sort of with my own ideas with this is that one of my sort of big things that I'm a fan of is infrared light because I think it sort of
Mm.
is a way to exert influences on tissue that modulate things like circulation and hypoxia through
Yep.
like a non-local effect because it's such a long wavelength that it's sort of at that sweet spot where it can penetrate several inches into tissue in a lot of cases without actually having to go and try to find like a local effect. And I suspect that with talking about things like bioelectricity, you're probably thinking along the lines of like ultrasound or other things could be like the best way to open latches in like a concentrated region of tissue. Um, but something else with infrared light, for example, um, so I was finding this sort of stuff on post-translational modification, and sort of the question arises like, well, can we non-locally break these bonds and sort of like reset post-translational modifications? And infrared light and other frequencies of light seem to be like the perfect tool to do that in some sense. And
Fascinating.
with nitric oxide pooling, for example, like the nitrosylation of different proteins, the bond can be broken by like a handful of different frequencies, and it's basically blue light, UV light, and interestingly, infrared light. And so there's this idea that like infrared light therapy dilates blood vessels and has this effect where it like expands blood flow to different regions. And there's different reasons that that might be the case. Some of it might just be that it improves oxygen usage at these different heme proteins and things like that. Like nitric oxide synthase is a heme protein. So that's one case where it might just be kind of a simple part of the equation of like, oh, it just improves anything with heme in it. And that's nitric oxide and the electron transport chain and everything. But my counter to that would be, well, maybe it's also just very broadly like exciting these bonds with nitric oxide. And when you do a certain duration of time, like you sort of reset nitric oxide pooling to some degree. And that nitric oxide can be maybe rapidly mobilized into the bloodstream to the
Mm.
point where you'd actually see like an increase in vasodilation. And so I'm kind of curious, maybe if you've thought about it from that standpoint, or whether there's other things that you've thought of using like to target latches opening. Um,
Yeah. Yeah, fascinating. I mean, I think there's a lot to that, and I do think that uh, even like there are many ways of getting energy into tissue, and there's just like uh, physical heat. There's infrared light. There's ultrasound, and I think like these are not the same, but their effects seem to kind of rhyme sometimes.
mm,
And so yeah, uh, super interesting. I think that just in terms of like interesting ways to perturb the system that may lead to opening latches, like like one strange claim that I'd say, okay, well, it feels weird, but it's not crazy. Uh, is something like either HIIT workouts or
Mm,
inversions, like headstands before meditating.
yes. Yeah.
I mean, like inversions, they do a lot of sort of interesting, hormone release. That it's like not a simple thing. But
Yes.
then
Yeah.
also, yeah, then just the mechanical, like, okay, let's invert the body, blood, goes down and sort of applies, more hydraulic pressure to potentially very heavily latched areas. And then you stop the inversion, you meditate. I expect big effect size, like surprisingly big, from things like that. And I guess like from a um, kind of a mad scientist, Ray Pete, meditation interested perspective. Like if you take a look at the things that the Buddha would be doing, he tried everything. It's like
Yeah.
he was, a real daredevil of his time. And so I think that he would be very creative today, in the same sense. And, not saying that, these are the highest probability interventions or whatever, but I do think that we can be a little bit bold and fresh with like we kind of we know what a meditation retreat is, but we could take a fresh look at that and try these things.
Yeah, definitely. With the inversions, I wanted to say, I don't know if maybe you've seen this before, but he's like a repeat adjacent blogger. I think his name is like Vaishna Vedanta or something like that. But he has an article or a blog called Heart Hormones, Inversions, and Immortality. And he talks about almost exactly this. He's like a yogic practitioner that got into repeat. And
Amazing.
the whole article is about basically how when you do inversions, it can increase natriuretic peptides, which are also increased by like exercise and higher sodium intake and things like that. And they can actually like lower blood pressure and have these different health effects. And basically, there's this whole other side of the equation of like you can get a lot of these benefits from things like just moving your body in like unusual ways. And it also kind of reminds me of like the idea of like scalp massage for hair loss, where people
Oh yeah.
sort of attribute that to saying like it's an increase in blood flow, which I think is huge. Like I think hair loss, like in the short term, the scalp is like under a flux of hypoxia. That there's a lot of these studies that basically show that like short term hypoxic sort of signals will stimulate hair growth, but I think in the long term you see these like issues like
Yep.
calcification and and sort of like prolonged hypoxic signals or inflammation is another driver, and that can basically create like a state where there's a prolonged issue with blood flow. And so maybe this brings in the interesting kind of thought too of um, maybe you could solve hair loss with latches in an interesting kind of way.
Sure. I mean, so I gave a talk last year, and just kind of simple sentences, and then I would describe what it meant to me and on Vesicomp. And one of the slides was everything is spasm.
Mm,
Uh, and I think like latches and spasms, there is this substantial overlap. And yeah, I guess like with hair loss, I wouldn't be surprised. I also think that the lymphatic system also has the same smooth muscle, and so you could
so one of
have
my questions
lymphatic.
for you actually was, do you think this applies to lymph? Yeah.
Yeah, 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 yeah. So it feels like, yeah, possible, very possible. Um,
Mm.
yeah,
So another one, are you familiar with the primo vascular system?
yeah, yeah. Yeah.
That's
Do you want
another
to
one that
say
I wanted to ask about. Have you seen anything with this I mean, maybe I haven't looked at it in a while, but I don't remember if the structure includes smooth muscle. I assume it does.
yeah? It's been a while, or like I learned about it in a context where I was not thinking about Vasocomp, so I haven't sort of refreshed my priors on that.
Yeah, that's interesting.
But yeah, it'd be super interesting.
Yeah. Yeah. Any last questions or any last thoughts or anything?
Yeah, I mean, I just I think that um, my my like part of my angle on this, as we said on before we started recording, just like it's fun to have surface area on all these different topics. Um,
Yeah.
and like I guess I'm especially bullish on this surface area or like overlap between, okay, we have like bioenergetics and meditation, Vasocomp stuff. And then yeah, I mean it sounds like the stuff, the imprinting stuff is also super central. And yeah, I
Yeah.
guess I'm like it's always a question of what like stable conclusions can be drawn, what interventions can be figured out, like what is the right sort of output? What are the right sorts of artifacts and stuff from this inquiry? So I don't necessarily have all the answers there, but
Yeah. Yeah. Maybe we can follow up at some point when you've had some more time to look at this. I'd be very curious to see kind of what other theories come out of that, like new angle of looking at things for you.
that'd be great.
All right. Fantastic.